Good morning, good afternoon, and good evening, everyone. My presentation topic today is about the regulatory uses of in vitro release testing studies on complex generic ophthalmic injectable.
Dr. Quinn, we see your other screen.
Okay. This one. Can you see my screen?
Yep. If you make it big, that'd be great.
Okay. So you can see my screen, right? So it's the PowerPoint screen.
Yep, that's the correct screen.
Okay, thank you. Yeah.
If you can make it full size, that'd be great.
Yeah, so this presentation will reflect the views of myself, should not be constituted to present FDA's view or policies. So here's the outline. In this presentation, I will first talk about the application of use in vitro release testing for bioequivalence purpose. I will provide some example about using IVRT to support the BE for complex ophthalmic products and complex injectable and inserted products. And then I will talk about use IVRT for quality control purpose. Here I will briefly talk about some general consideration when you use IVRT for QC purpose. I will also talk about the other application of IVRT, which is used for IVIVC. In vitro release testing, they are very useful in vitro tests that used to measure the rates and extents of dissolution of the drug from products under specific conditions. And if it's successfully validated, an IVRT method should be discriminated to the changes in physicochemical changes of the drug products. And it can also to use as a quality control tool to monitor drug stability as well as consistency of manufacturing process. So one of the most often used application for in vitro release testing is to demonstrate bioequivalence. It can provide information about the in vitro release rates or profiles between the test and reference products. When used for BE purpose, oftentimes it serves as one component of the totality evidence approach, which means that so in most of the case, IVRT is used in conjunction with other categorization or in vivo study for the BE purpose. When used for BE purpose, IVRT is used as part of an in vitro only BE approach or used in conjunction to an in vivo study to support BE. And also, it can be used to support waiver of in vivo B study for other strands. In the next few slides, I will provide you a few examples about how IVRT can be used for support of bioequivalence. Those examples actually are from the product service guidance made for those complex products. The slide showing here provides some example about the complex ophthalmic drug products. As we can see here, there's a wide range of ophthalmic products, including ointments, emulsion, gel, and suspension products. For all of these products, the in vitro only B approach is recommended in the product service guidance. And IVRT, it serves as a part of the in vitro only B approach. So this is a more detailed example about cyclovir ophthalmic ointments. This product service guidance recommended in August 2021 recommends using in vitro option to demonstrate a bioequivalence between the test versus the RD products. And as a highlight here, the in vitro drug release test of cyclovir from the test and reference standard products can be used as part of the categorization to support a bioequivalence. This is in conjunction with other physical chemical categorization and also the precondition that the test product in IRD should be qualitatively and quantitatively the same. This slide shows some examples about the in vitro release testing for the purpose for the complex injectable products. Here I have some examples about nanosuspension products such as meloxicam, dantrolene sodium, azacitidine. I also have examples about some of the injectable suspension like penicillin G, benzothine, triamcinolone, acetone, as well as some other in situ foaming products, as well as intravenous emulsions. For those products, the product specific guidance has in vitro only the approach available, and IVRT is used as a part of the in vitro options. Here's some more example about use IVRT for complex injectable to support BE. So in these cases, I have some example about several liposomal injection products and albumin bound nanoparticles, as well as risperidone PLJ microspheres. So in these cases, the product service guidance does not have in vitro only the options, but still IVRT has been used in conjunction with in vivo studies, such as a clinical endpoint, such as a PK endpoint study to support the bioequivalence. This slide shows some examples of complex inserted products. Here we have levonorgestrel intrauterine device and some vaginal inserted products, as well as copper intrauterine device. So in these cases, the IVRT it's used in conjunction to either an in vivo ex vivo study, a PK endpoint study, or a comparative clinical endpoint study to support BE. This is the PSG page of the levonorgestrel intrauterine device. So this PSG recommends 2 studies, in vitro and in vivo ex vivo studies. When certain criteria are met, the formulation with the Q1, Q2, the same to the reference products, have equivalent physical and mechanical characteristic to the reference standards. You can use comparative in vitro drug release study for the intended period of product use, which is 5 years, in conjunction to in vivo ex vivo clinical study to demonstrate bioequivalence of these drug products. Here's some other application for in vitro release testing. It can also be used to support the waiver request of in vivo testing for additional strengths. Here I provide one example, which is polyperidone palmitate extended release injection. This is a long-acting injectable products for antipsychotic use. This product is available as 5 strengths, and the PSG recommend that you can, if you use the 156 milligram strengths for the bioequivalent studies, for other strengths, you can apply a waiver request of in vivo testing based on acceptable bioequivalent studies on the 156 milligram strengths, acceptable in vitro dissolution testing of all strengths, and proportional similarity of the formulation across all strengths. So to satisfy the PSG requirement here of acceptable in vitro dissolution testings, one key aspect that you need to compare the dissolution profiles across all the test product of different strands, which means that you may want to, for example, use F2 similarity factors to compare these strands, the test strand that it's not used in the bioequivalence study versus the strands used for bioequivalence study. Here are some considerations for in vitro release testing when used for bioequivalence purpose. One key aspect is that when used for B purpose, an in vitro release test methods should be able to discriminate the batches of product that are intentionally made to be not bioequivalence. This can be achieved by using the product that manufacture with meaningful or intentional variations in critical quality attributes of the drug product. For instance, for injectable suspension product, if you identify that the particle size is the critical quality attributes, you may consider using a product with different particle size to demonstrate the discriminate power of the dissolution methods. And the other consideration, as I think Dr. Kozak also mentioned in his talk, is that when IVRT is used as part of totality evidence approach, it's generally not, it's desired, but it's not expected or required to correlate or be predictive of in vivo bioavailability. And we talk about the discriminative power of the IVRT methods. There's some, in some cases, for example, for product that is intended for rapid dissolution after injection. One example is dantrolene sodium, which is a nano suspension injectable products. This product, once injected through the intravenous administration, it gets fast dissolved. So the IVRT for those type of products is aimed to show comparative fast release. It's acknowledged that in such cases, discriminated power may not be practical. So for those type of scenarios, you may consider including proper justifications when you develop the IVRT. We just talked about IVRT for B purpose. Now let's talk about its application for QC purpose. IVRT is a very important tool for QC purpose. It can use to monitor the drug stability and consistency of the manufacturing process. It can detect, if it's properly validated, it can use to detect variation due to product manufacturing, to detect changes during product storage that may impact the product performance, as well as to support minor or moderate CMC changes. So when develop an IVRT, like as we mentioned here, right? So for most of the complex products, like ophthalmic, complex ophthalmic products, complex injectable or complex inserted products, there is no compendial IVRT method available. So in this case, you may consider develop appropriate product-specific methods for the intended products. You can also check whether there is a method published in USP or available in FDA database. If the answer is yes, those methods may serve as the starting point for your IVRT method development. Here, please keep in mind that those methods published in like FDA database, they are non-binding. So those can be used as a starting point. You may modify the methods based on during your development. So regardless, even if there is a method published in FDA database, you still need to properly validate if the method is suitable for the intended use. Here we can talk a little bit about the IVRT acceptance criteria. Here are some considerations. The complete in vitro drug release profile data for bioequivalence and registration batches should be used to set the acceptance criteria for in vitro release testing. And in general, a minimum of 3 time points may be used to set acceptance criteria for extended drug release products. And these time points should cover early, middle, and late stage of the drug release profiles. So the last time point, generally, you should have at least 80% of drug release. If for certain products, the maximum amount of drug release cannot achieve 80%, you should demonstrate that the last time point has been reached the plateau of the drug release profile. So in general, the selection of drug release acceptance criteria range is based on mean target value plus minus 10% and greater than 80% for the last sample time points. Wider criteria range may be acceptable if proper justification is provided. Examples including successfully established an in vitro in vivo correlation, or you use physiologically based PK model as supported information. So if you have any question related to your dissolution development, you can always submit a control correspondence or pre-ANDA meeting to FDA to get FDA's comments and input. So when you submit those inquiries, please specify that your IVRT is for BE purpose or for quality control purpose. You can get the target device. You can get a target device from FDA. So regardless, an IVRT is used for BE purpose or used for quality control purpose. You should have the in vitro drug release method development report and validation report to support the selection of the method you selected in your ANDA submission. In general, those reports should contain information including the drug solubility profile data, detailed description about the method you use, especially for non-contending method. You should have very detailed description about the apparatus you use, as well as sample introduction methods and some other information. And also, you should have detailed description about the development parameters supporting the selection of the proposed method as the optimal test. And you should submit complete dissolution data and also data to support the discriminative ability of the method you select. In addition, you should provide supported validation data for the method, including specificity, precision, accuracy, linearity range, stability, and robustness, etc. Now we can talk a little bit about another application of in vitro release testing. Actually, this application is also relevant to either bioequivalence or for quality control purpose. This is, you may use in vitro release testing to establish an IVRT. FDA already published guidance for industry regarding IVRT in 1997. Although this guidance for industry is for ER oral dosage form, the framework and general principle of this guidance may still be applicable when you develop IVRT for those complex ophthalmic, injectable, or inserted products. If an IVRT is successfully developed, you may use that to set dissolution specifications or use that serve as surrogate for an in vivo B study. So for generic drug development, an IVRT may be used to support the use of IVRT as surrogates for in vivo B studies. So it's for non-oral complex drug products, it's often very challenging to establish an IVIVC, especially level A IVIVC. So in these cases, if you want to use IVIVC to serve as the surrogate for the in vivo B study, it may not be easier than conducting in vivo B study. So alternatively, when an applicant explore in vitro alternative B approach, you may also consider using IVIVC to support your proposed in vitro alternative B approach. So in this case, the main purpose of the IVIVC is not used to serve as surrogate for the in vivo B study, rather than used to identify the CQA design space. Level C IVIVC may be acceptable in such scenario. I would like to use this slide to conclude my presentation. IVRT is a very useful tool to support the regulatory approval of those complex products. We talked about the IVRT used for DE applications. It can be used as part of the in vitro only approach, or used in conjunction with in vivo B study to support it. It can be also used to support the biowaiver application of non-biostrands. And another often application for IVRT is for QC perspective as well. Applicants should submit sufficient information to support that an IVRT method is appropriate for its intended purpose. With that, I would like to conclude my presentation. Thank you for your attention. So I would like to introduce our next speaker, Dr. Mark Hallas. He's an associate director working within analytical development in Teva's combination products and semi-solid group. Thank you for presenting, Dr. Hallas.
Okay, thank you. So I guess there's a tiny bit of overlap between the previous presentation and this one, but I'm presenting on the role of the in vitro release test in generic drug development. As I said, I'm working in the combination products and semi-solids group in Teva Larne. So my team is responsible for developing the full range of analytical tests for many of the complex products that we're developing. Across my own career, I've worked with solid oral dosage forms, semi-solids, implants and injectables, all in support of regulatory filings. Just about also on myself, when I'm not in the office, I enjoy walking through some of the fantastic scenery we have here in Northern Ireland and spending time out on the water. We're very lucky to have such fantastic resources around us, and of course the family dog likes to get involved too. So in vitro release testing, as we saw in the previous presentation, the in vitro release test, it's simply a laboratory test to measure the rate and extent of drug release from a drug product under specific test conditions. And I think the important thing there is that the rate as well as the extent, that the test that's being used must be capable of discriminating. It's always the word you hear when you're talking about dissolution. So why we need an IVRT? I would say it's critical in supporting the regulatory filing, and rather than being significant. But it doesn't just start with the filing. The the in vitro release test starts from the very first batches of your product that are made, and an appropriately developed test will help the formulators develop a formulation and a process that meets the drug release target that you're aiming for. Once you've got that far, the same test under the same conditions as we've seen from the definition will be able to help you compare the release profiles from your reference products or different in-house batches in advanced clinical studies to try and get an understanding of how you think those products are going to behave in vivo. And of course, then once you've made it through the clinical studies in your file, that same method supports the product through the rest of its life cycle. There's plenty of regulatory guidance and publications there to help direct you what is to be expected from your drug release method, but it really can be distilled down to that you have to submit a report that justifies all the parameters of your test and shows that your test is suitably discriminating. And there's a slide I pulled from an old FDA presentation of some common deficiency, and I think there's a presentation later on going into that in more detail. The dissolution method development must be included in the application. The data must demonstrate that the method is discriminating, and you must have data to support your acceptance criteria or your Q. Although for the complex products, there's not an awful lot out there in the way of guidance. The guidance from the oral dosage forms and extended release kind of serves a purpose to help direct us the right way anyway. And you can see a little snippet from that a discriminative dissolution method is preferred because it's indicative of possible changes in quality. In the extended release, again, once a discriminating system is developed, dissolution conditions, test conditions should be the same for all the formulations tested for the bio study, to make sure you have that equivalence and understanding of the product that you're working with. On the Lawrence site where I work, our approach is similar to Dr. Chin presented. So we always look for, see, is there a USP method published, and evaluate that for its discriminatory power with respect to the formulation that we're working with. If that doesn't work, it's not discriminatory, we look in the FDA dissolution database, and again, trying to establish discriminatory power. And then, of course, if there's no USP method or FDA dissolution database method, or neither are discriminatory, we have to develop a method in-house. It doesn't necessarily have to be from first principles. There's literature out there, there's quality review, there's summary basis of approval for generic products that would give you an idea of the test conditions that maybe the reference product has been using, that can certainly serve as a stepping off point for your own method development. But as we said, the conditions that you end up with still need to be justified. And what you need to justify, as we probably know, is you need the apparatus that you've picked, whether that's compendial or non-compendial, the media volume and composition, how you're agitating, what that rate is. If it's a permeation test, that your membrane is it's not interfering, it's not rate limiting, what your sampling points are, and then of course ultimately those all go together to pick your specification. But as it's been mentioned, and the word that keeps coming up, is that the aim of the release test is to be discriminatory with respect to meaningful changes in critical process parameters or excipients. And this is really important for the method to fulfill its role in supporting your product development and further through the life cycle. It has to be discriminatory, or you're not making decisions based on the right information. So the parameters that we would look at in terms of assessing what would be a meaningful change from a formulation, really summed up there on the slide. We look at material attributes, formulation variables, and process parameters. So those material attributes could be from your API particle size, API, the form of the drug, formulation variables and the relative ratios of components if you're not going QQ, or indeed on the grade of some of those excipients and whether that could impact the product at the end of the process. And then of course the processing parameters. This isn't my area of expertise, but there will be people within your structure that understand how these factors could impact the output, impact the finished product, and those people and yourselves should come together using the tools that are laid out in ICH Q89 in terms of risk assessment of the product, and helping using this quality by design approach to understand the risks as you move through the process and identify how those risks can affect your critical quality attributes. Now, we'll focus here today obviously on the drug release from or IVRT. But what we do is walk through the formulation, walk through the process, and identify and assign a risk to each step as to whether it could impact the drug release test. So this is an example pulled together by one of our project teams for a capsule product. So we have the CQAs, critical quality attributes across the top, description, assay, antioxidant, drug release, water content, and so on. And this isn't the full, it's just a snapshot, but down the side, you can see there's a list of the excipients and then some of the processing steps. And if you look at the dissolution column, you can see that this risk assessment has identified that the viscosity modifier could have an impact on dissolution. Surfactant concentration, or the surfactant itself might have an impact. Hold time during manufacture could have an impact on it. And all of these need to be, or this information can be used then to help develop those batches that we can use to challenge the dissolution method's discriminatory power. It always comes up about how much do you change any of the components or change any of the processing parameters. For me, meaningful change in composition is plus or minus 10%. That is probably outside the window of the tolerance of your manufacturing site's capability. So you wouldn't expect the normal product to be plus or minus 10%. So that I would think is considered meaningful. For processing parameters, it's really on a case by case, and it needs your subject matter experts to weigh in to say whether the effect of temperature, the effect of hold time, stirring speeds, whatever would have a potential impact on the finished product. So once we've drilled down into that and manufactured batches that we would expect to be suitably discriminating, we can start to test them. So this is just an example of a product where we changed the viscosity modifier in the fill. And as it's been mentioned in a couple of presentations already, this similarity factor model independent test is used quite a lot just to compare the profiles. There are some limitations to this test, and certain criteria have to be met. You must have 12 units per product. You have to have the same time points. You must have 3 time points and only one of those when there's more than 85% of both. And your product the results of the test need to be really repeatable. And then we apply the equation from the guidance, and it'll give you a value between 0 and 100, with less than 50 being not similar and greater than 50 being similar. So we can see from this that changing the viscosity modifier in that particular formulation gave us an F2 less than 50. So we could say that our method that we used in this case was discriminating for the viscosity of the capsule fill. The same test is used to compare profiles of test and reference prior to the clinic, to give a wee bit of confidence that going into it, your products are going to behave the same. I know it's not IVIVC exactly, but the more similar they are on in this release test, that it gives you that confidence going in. In this case, that the F2 was greater than 50, and we would probably be happy enough to go into a clinic with that. This example, and again, Dr. Chin touched on it at the end of his. This product has greater than 80% at 5 minutes, greater than 85% at 10 minutes. In this case, the F2 value is not applicable. It doesn't meet the requirements of this test. We don't have 3 time points above 85%. And when you have a formulation like this that is really fast releasing, and you've the data to show that you've investigated the agitation rate, the media, all the things that were in the slide above, and tried to retard that release as much as possible. And if you can't do that, then there's definitely a position that perhaps drug release rate or dissolution is not a critical quality attribute for that product. And there's a discussion then between yourselves and your regulatory department about how you approach FDA to talk about that, because what you don't want to do is go in without all the right data supporting your final specification and your drug release method or not, if you don't need it. People pick up USP methods, FDA methods, and use them directly, and it is a very, very risky strategy. It may work for you, but without challenging the discriminatory power for your product, you could end up going into a clinic and where the products just don't release the same, and that is a very costly exercise. So there's a lot of money spent when it could have been avoided. The other risk of not showing your discriminatory power is this risk of a major CRL or refusal to receive if there's no development report. So the in vitro release test is really critical in getting you as far as that clinic. So, as I say, a properly developed test will support your formulation process development. Through analysis of your experimental batches, you can demonstrate that your formulation is robust, that your process is robust. Release profile target for the reference or whatever it is that you're testing against. And of course, sometimes there needs to be a bit of back and forward between the two to hit that sweet spot where you've got the right release profile, but your process is robust. There's no point in really having one or other moving forward into the commercial sphere, because your operations group won't thank you for having a process that isn't robust. And your IVRT will definitely help support that. Once you have your formulation and you've released for clinic, it's been discussed before, and there's more slides or presentations later on going into this in more detail. But with an appropriate discriminatory test, you can use the clinic data to develop an IVC model. And if it turned out maybe that your product wasn't equivalent in a clinical study, that model can be used to support further product development. As was said, for these complex long release generic products, developing an IVC, a robust correlation could be very challenging, but it can be indicative. And we have had success here where we were able to look at the blood profiles from the clinic and look at our...
Mark, sorry to interrupt, but we've lost your screen.
Oh. Is it back? No.
Yes, it is.
Sorry, how far did it get? When did you lose me?
I think right there, we were able to hear you the whole time, so no worries.
All right. So we got that. So of course, then support for the clinic, and then using those, the blood profiles, your in vitro profiles, to have an indicative model that'll help you develop your product a little bit further, so you can go back into a clinic with more confidence that you'll pass it. Or the other option is, as Dr. Chin mentioned, supporting bio waivers and having the appropriate method for that. And for each individual product, there is in vitro bioequivalence. There's product specific guidance. There's guidance for industry. There's draft guidance out there. You'll just need to go and look for them to see if they exist for your particular API and dosage form that you're testing. And then, of course, the final step is getting through approval, and that the in vitro release test will support your product through the release and stability testing of your pivotal or registration batches, and on through the product life cycle, to make sure that you've got batch to batch consistency, that the batches aren't changing over the course of their shelf life, as we've talked about before. The method also is used to support any post-approval changes that your operations group might be thinking of. Because you know that method is suitably discriminating, you're able to look at whether it's a change in an excipient supplier or a change in a piece of equipment during the manufacturing process. They'll be able to meaningfully evaluate whether that change is likely to impact the drug release rate from your product, and therefore, is there any risk to your patients. So just in summary, the test has a critical role in drug development right the way through the life cycle. I think it's really important in aiding the formulation and process design to make sure that it meets the requirements that you need in terms of release profile, but also that formulation and process is robust through the risk assessments of all the material formulation and processing parameters that are likely to impact it. Of course, your test supports the release to clinic, and that's really important to mitigate any patient risk. You want to make sure you're not going to dose dump or anything of potentially dangerous chemicals, but it also provides a degree of confidence beforehand that your test and reference will be similar, just to make sure the clinic's going to pass. And once you get through all that, through the product life cycle, so it is really important. It's hard to underestimate the importance of it. I think when I first started, the in vitro release test was really seen just as it was a performance test, but on the finished product, that was just part of the specification. And the importance of the in vitro release test has been developing year on year, and more guidance is out there about how the method needs to be developed and how it needs to be justified. And we saw from Darby's comments earlier that it's going even further to, try and make it more bio relevant and more meaningful for the dosage form that's being tested. And it's really interesting, really exciting to be part of that and going into the future. But it's just what it says on the screen there. So it's getting through, supporting your product development, supporting going into clinic, and then the final product life cycle. And that's me. Thank you.
Thank you, Dr. Hallas. Our next speaker is Dr. Yunjin Park. She served as team lead for division of bioequivalence, Office of Bioequivalence in Office of Generic Drugs at FDA. Thank you for your presentation, Dr. Park.
Thank you, Bin, for the nice introduction. Can you guys see my presentation slide?
Youngjun, you're still in the split screen.
Okay, let me try this. So I have a little bit hard time to.
That's good. Just the need full screen presentation mode.
Yeah, looks like I cannot see my. Guys. Sorry for the technical difficulties here. Let me close again and then. How about now?
We see it just needs to go into presentation mode.
Okay, got it. Is it okay now?
Yep, looks good. Thank you.
Okay, so sorry for that the delay. So I present the bioequivalence consideration for IVRT method for ophthalmic product. So this presentation reflects my own view, not represents the FDA views or policy. So today's talk will start with a general BE approach for the ophthalmic product with IVRT recommendations in the PSG, key parameters of IVRT, and common BE deficiencies and consideration on IVRT method. So far, more than 60 PSBs were published for the ophthalmic product, and approximately half of them are solution products. So today's talk only focuses on the non-solution product with comparative IVRT recommendations. So this pie graph shows the number of the PSD of non-ophthalmic, non-solution ophthalmic product, 18 PSGs for the ophthalmic suspension, 3 PSGs for the ophthalmic immersion, 2 PSGs for the gel product, and then 7 PSGs for the ophthalmic ointment are available now. In general, the PSG gives in vitro and in vivo options, typically the Q1, Q2, Q3, and comparative IVRT were recommended for the in vitro option. Clinical endpoint study or PK endpoint study in aqueous tumor were also recommended as an in vivo option. There are some exceptions. So the PSG only recommend in vivo BE study without in vitro options such as retinoprost ophthalmic immersion and brinzolamide ophthalmic suspension. These drugs indicated for the treatment of the glaucoma by lowering intraocular pressure. For these products without supporting in vitro and in vivo relationship, agency believe in vivo BE approach is more appropriate to reduce safety and efficacy risk. But there are also some products currently PSG only recommend in vitro study. For example, the low strength of loteprednol ophthalmic suspension and cobramycin ophthalmic ointment. The PSG only states that in vitro studies, but if your product is not Q1, Q2, applicant may contact the agency in advance to discuss the drug development plan. Yeah, we already heard the role of the IVRT method, so we can skip this slide. And this is the list of the drugs agency recommend comparative IVRT method and approved drug product using in vitro option. IVRT method were recommended total 9 suspension products, 2 emulsions, cyclosporine and dipropionate, 1 gel product, and then 4 ophthalmic ointment drugs. Some drug product has multiple PSG because of the multiple reference product. Agency approved 4 ophthalmic suspension products like dexamethasone and tobramycin ophthalmic suspension, or rotiprednol suspensions, and 1 cyclosporine and 2 dipropionate, and 1 rotiprednol ophthalmic gels using the in vitro option. So what does this PSG say about the IVRT? The PSG used these standard languages. Acceptable comparative in vitro drug release of API from the test and reference formulation. The methodology used for IVRT testing should be able to discriminate the effect of process variability in the production of test formulation. Detailed information on development and validation of a proposed IVRT method should be provided. So what can we learn from this simple statement, as Dr. Kojak and then Bin already talked about it? There is no compendial method or general guidance for ophthalmic product. So, but agency recommend to develop the product-specific IVRT method. An IVRT method should be accurate and precise, sensitive, and selective to ensure the equivalence in drug release between test and reference product. And when used in part of totality of evidence approach, no need to be limit physiological conditions or performance. So from now, we can take a look about the key parameters of IVRT method ophthalmic product. So in the method development stage, the first step may be the selection of apparatus and adapters. This slide summarizes the commonly used apparatus: USB apparatus 2, paddle, USB apparatus 4, flow-through cells, cortical diffusion cells, and rotating bottles. These apparatus usually used with various adapters or sample holders. So USB 2 is quite versatile apparatus with a different combination of adapters, such as dialysis membrane, immersion cell, and then suspension cup. And USB apparatus 4 usually used for the low solubility drugs with the flexibility of flow rate changes and unlimited volume of media if you use the open loop. Cortical diffusion cells also can be used, but I don't see that many cases for the liquid phase ophthalmic product using the vertical diffusion cells. And we have seen a couple of cases of rotating bottle with dialysis membranes. So usually the applicant tried several apparatus and demonstrate that their choice of apparatus and sample holder were optimized. So what other factors we can consider is membrane. Membrane is just for the usually used for the separation of sample from media. The applicant should provide the comparability of this membrane with media and also no diffusion resistance or the sample adhesions. For the receptor solutions, solubility of API should be submitted to support sink condition, and stability data also need to support the sample stability in media until last sampling time. The pH of the receptor solution and temperature also should be considered when you develop the IVRT method. When also often we see the applicant didn't submit the optimization of loading amount of ophthalmic product. However, sometimes the sample loading amount can be a critical factor, so you also have to be considered. For the sampling time selections, basically the complete release recommended for ophthalmic product. So depending on the apparatus and sample folder, the sampling times can be up to a few hours, or a few days. So if your IVRT method cannot make a complete release, we expect some justification, such as the impact of incomplete release on bioequivalence evaluation. For the method validation stage, there are many factors we have to validate, but the most important 2 areas we can work on it is the reproducibility and discriminatory ability. So IVRT method should be precise with a low percentage CB and the robust against minor disturbance, for example, flow rate, temperature, sample loading amount, dissolution medium concentration, pH of dissolution media, or media volume. It is not necessarily tried to all these examples, but applicants should submit that evidence the IVRT method is robust. So IVRT method should be able to discriminate non-bioequivalent batches. So sensitivity of the IVRT method can be demonstrated using different amount of API, and specificity using non-bioequivalent batches with process variability, such as the changes in particle size, changes of excipient can be demonstrated discriminated. So currently FDA does not have any kind of guideline about the change of the degree of changes. Yeah, for the example, the different API amount, we often see the plus minus 20% or the plus minus 50% changes, but that changes should be reasonable in range. For the pivotal IVRT study, we expect a complete and comparable release. You can see the left side graph to the acceptable cases, test and reference comparable release, and also it shows the release was reached to the plateau. So it's the example of the acceptable, but right side graph is not acceptable in terms of this incomplete release. So you have to work on it. For the statistical evaluation, we already heard several times. Generally, we recommend this similarity factor approach. But if you cannot use this approach, you can also try other statistical method with appropriate justification. So let's talk about the common deficiency. The first is missing document. As a regulatory agency, we expect a complete application package containing the proper documentation. For study report, we often see the IVRT method development report is not submitted, and sometimes method development is part of the method validation, but ideally we expect the independent report for the IVRT method development. Assessors also need to see the raw data to confirm the applicant's study result. And certificate of analysis or batch information of all drug products, including altered formulations, should be submitted containing these information: manufacturing date, manufacturing process change, batch size, potency, and content uniformity if applicable. And all SOP mentioned in the study report and study protocols for the method validation and pivotal IVRT study also should be submitted. The proper documentation helps us to reduce the assessment time. The second is the missing information and justification. Even though the full study report was submitted, we still see the lack of supporting evidence to optimize method. We expect to see the optimization step, how you select your parameters, such as your choice of the membrane or dialyzer, and both sides of membrane, selection of release medium, temperature or pH, and so on. The rationale behind the selection of parameters are important to understand the IVRT method. For method validation stage the assessors frequently see the incomplete validation, for example, missing solubility and stability method, and mass balance, the robustness data. Sometimes they didn't submit any sensitivity data to support discriminated power. And also the analysis method for IVRT sample should be validated. For IVRT data, the high variability is one of major deficiencies across the different ophthalmic formulations. So we typically recommend 12 units per batch and 3 batches per test and reference product. The left side graph showed high unit to unit variability. So around 20 hours, the maximum difference of the release from between the units almost 40%, which brings a question about your IVRT method. Right side graph shows the high batch variability. One batch is quite different with 2 other batches. Then we have a question about your IVRT method is under control or not. For discrimination, These are example of excipient change and the manufacturing process changes. In the left graph, the applicants changed the amount of the one of excipient, but the drug release was not discriminated. This might be because the specific excipient is not critical for the drug release, or the degree of the changes may not be detectable using current IVRT method. The right side graph is an example of different global size batches. So applicant manufactured the lower global size and high global size batches, but in IVRT method did not discriminate lower GSD batch, but high GSD batches showed slower release. So in case your IVRT method cannot discriminate on certain changes, yeah, you can submit the justification. So key consideration of IVRT method in different ophthalmic dosage forms. So far, the majority of the application we received for ophthalmic product are emulsion, like a cyclosporine and diclofenac. Agency and industry was struggling in the beginning of this ophthalmic emulsion product development and assessment. But based on the accumulated knowledge from the industry and an agency, we recently approved 3 ophthalmic emulsion products. So one of major concerns of emulsion products are membrane selection and mass balance study. The proposed IVRT method is measuring drug release from the formulation rather than the transfer of the oil droplet across the membrane to media. So simply say the pore size of membrane should be small enough to hold emulsion globule, but big enough to release API. The complete and comparable release should also be considered when you develop the method. And the evaluation method of drug release data using semi-solid transdermal product is not applicable to an ophthalmic emulsion product, considering the different release mechanism from misleading boundary and emulsion globule. And for the ophthalmic emulsion product, release profile are compared using the similarity factor. For ophthalmic suspension product, it's a little less complicated than the emulsion product, but the majority of these deficiencies was found out the discrimination power. The particle size of ophthalmic suspension is an important consideration in terms of the irritation and bioavailability. So we expect the discrimination of different particle size submitted. You can also think about the process parameters change, like a homogenization, and also the excipient change. And we sometimes see some high variability issue about the IVRT method with the sample kind of loading technique or the amount of sample loading for suspension product. So far, agency only developed the 1 PSD, or the 2 PSDs for the roteprednol etavonate ophthalmic gels. And we have a few applications using in vitro options. But the gel product shares some common critical attributes with suspension product, such as particle size. So discrimination of IVRT method for different particle size or the viscosity is key parameters. And also you can think about the pH, osmolality, or the excipient changes to support your IVRT method discrimination power. So the in vitro B approach of the ophthalmic product are relatively new for the agency. So we are also learning the knowledge and information from scientific communities. So today's presentation is based on our current knowledge. So I like to point out that these may be taken home message. The first one is the understanding the purpose of IVRT method for bioequivalence. To confirm the proposed generic product with a comparable release rate to that the reference product to ensure deliver the drug in the same manner of this reference product. IVRT method, yeah, reproducible and discriminated. And second, I think it's important to understand the your ophthalmic product release mechanism or factors controlling drug release. And drug product specific IVRT method is recommended. There is no one size fit for IVRT method. And we see the various IVRT method was submitted. So, there is a room to you can work. And last but not least, the quality of the study report, it should be submitted to save time of the review cycle. So I'd like to wrap up my presentation with acknowledgement. I'd like to thank you to my team members working on the ophthalmic product, Dr. Sylvia Chumo, Dr. Li Chen, Dr. Jinnan Lee, and Dr. Jiangqi Anharia, and also my DB2 management, Dr. Hongling Zhang, Zhao Zhang, and Diana Vivian, and also my wonderful colleague from ORS, Dr. Yan Wang, and then the project, and Xiaoming Su from the OTR. Thank you so much for your attention.